Hey, Twelves, welcome back to our next video on photosynthesis. Okay, obviously we've looked at photosynthesis, so a quick recap. The main reason the plant does photosynthesis is to create glucose. Okay, we also looked at our two different stages of photosynthesis, so the light-dependent stage and the light-independent or carbon cycle. Okay, and how that produces glucose. Now we also looked at C3, C4, and CAM plants in the last video. All right. So we looked at the differences between different plants and how they photosynthesize so that it's effective. All right. Why do we do that? Because this carbon cycle here has an enzyme in it called rubisco, which is highly affected by temperature. Okay, remembering the co on rubisco is carboxylase / oxygenase, meaning it can work with carbons or can also work with oxygens. All right. So our C3 plant, it just does its basic photosynthesis, and the carbon cycle, or light independent stage, occurs in the mesophyll cell. All right, so if it gets warm, the photosynthesis becomes photorespiration, okay, and therefore inefficient creation of glucose. Whereas C4 plants, they separate anatomically or physically separate the mesophyll with a bundle sheath cell, and that's where the carbon cycle will occur. And the oxygen can't get through into that bundle sheath cell, okay, which allows the C4 plant to more effectively create glucose in a warmer environment. And then that CAM plant is in that extremely hot, arid, desert-like environment, and it has this temporal change, right, which means like a time-bound change, of which it changes when its stomata is open. So it's now open during the night instead of daytime. So that's when it fixes its carbon, all right, or collects its carbon, stores it as malic acid, and then during the day it closes its stomata so that it doesn't lose a lot of water in that heat. Okay, but it still has the stored carbon, and then it can do photosynthesis during the day in those mesophyll cells. All right, we're going to look at some factors affecting C3, C4, and CAM photosynthesis, and it should be some pretty obvious ones here. All right, so what are our main factors? So we have light availability, so the amount of light the plant has. The water availability, the amount of water the plant has. Temperature and or pH, okay. So hot, cold, acidic, alkaline, and of course the amount of carbon dioxide, so carbon dioxide concentration, okay. And as you can see in this graph here, we have light availability graph and rate of photosynthesis, and then we can see that there's different limitations depending on here's temperature, here's carbon dioxide, here's some other factor. All right, so that could be pH, or it could be water availability. So this light availability is the same. So really that allows it to always go to about here, and then from there it branches off. The rate of photosynthesis will increase to here if there's a high concentration of carbon dioxide and a higher temperature. All right, unless it's a C3 plant. Then this middle one, high concentration of carbon dioxide but a lower temperature. All right, and then low concentration of carbon dioxide and really low temperature. All right, so very interesting there. All right, so light availability, plants need light. It's pretty simple here, okay? So we get an increase in light, and then there's eventually a tapering off point or a plateau for the rate of photosynthesis. You can keep increasing the light after that. It's not going to photosynthesize any further because there is another mitigating factor, okay? So some other factors now affecting it could be carbon dioxide concentration, etc. The other thing we have to note about light intensity is, of course, the different wavelengths of light and how that affects photosynthesis too. So relative rate of photosynthesis, as you can see, white light, pretty low. All right, purple violet, good. Blue light, decent. Green light, very low. Yellow orange, decent. Red light, very good. Black, which is similar in a way to white, very low. So white and black and green, very low. Okay. We also have water availability. This is pretty obvious here because it is, of course, an input to the light-dependent stage, all right, of photosynthesis. If there's a lack of water, it is going to decrease photosynthesis. All right, pretty simple. And of course, transpiration as well. but that's more year 11. And of course, if there's too much water, the plant will plateau off at its photosynthesis rate. And then, of course, if it's flooding and stuff like that, the plant will continue to absorb and the cells will eventually burst and the plant will die. Okay. We have temperature and pH, two really, really important factors. Okay, especially temperature, because that really heavily affects Rubisco. And of course, pH will affect things if it's too acidic or, too alkaline. Unless it's something like an extremophile, like an acidophile, it will not be able to survive. All right, so you can see the graphs here. All right, rate of photosynthesis with the temperature graph, it kind of increases at a slower, slower at the start, and then it becomes a little bit exponential as it's going up heaps here. And that's, of course, the movement of molecules as the heat increases from really cold to warm. All right, and then you get to your optimal temperature, warm or cold, warm. And then as it gets hotter, this starts to drop off really fast. Okay, that drops off really fast because it starts to denature the enzymes. All right, if we look at this one, it's almost that perfect bell-shaped curve there, and that's pH. All right, obviously most things will be in that middle sort of area. It could be 7, it could be 6, it could be 8. It depends on whatever the optimal pH is for the specific plant. And then from there, the further it gets out of its optimal range, it will eventually become denatured. Okay, the enzymes, that is. All right, we also have our carbon dioxide. All right, and obviously plants take in the carbon dioxide through the stomata here, as you can see. All right, and that graph also tapers off. All right, so increases to a certain point, and then that will plateau because carbon dioxide concentration is no longer having an effect on the plant's photosynthesis, and another factor is affecting it. It could be sunlight, it could be light concentration, light availability, water availability, etc. All right, and these are all your factors together. So light, temperature, pH, carbon dioxide concentration, water concentration or availability, and enzymes are included too. If they're inhibited by different things, that can affect it as well, but that's less important. The important ones are these 5 here. Alrighty, we can also talk about CRISPR technology in this class. It's not really a factor, but it's interesting. What does CRISPR do? In plants, it can create an increase in crop yield. All right, so larger, bigger fruits, more fruits, etc. It can give crops different traits like abiotic resistances. All right, so resist to pests, etc., or like biotic resistances like that, or abiotic resistances, resistance to heat or frost or whatever, herbicide. It improves nutrients and fruit quality too. All right, but most importantly, you can improve the photosynthetic efficiency of a plant with CRISPR. Okay, so changing up those genes, cutting out one thing, inserting another thing with Cas9. Then you never know, you might be able to increase the efficiency of photosynthesis or decrease photorespiration or increase chloroplast concentration, whatever it is, to increase that effectiveness.